Hi, my name's Tucker Folscroft, and today I'll be presenting on biotin's role as an effector of glycogen phosphorylase B in the human body. So, biotin is going to be a very important molecule for a lot of processes. It mainly serves as a cofactor in a hexokinase, the process by which glucose gets converted to glucose 6 phosphate. And because of the variety of roles that biotin plays in the body, I was wondering what it would do to affect glycogen phosphorylase, the enzyme involved in the breaking down of glycogen to form glucose 6 phosphate. So biotin's going to be a B12 derivative, and it's going to be a water insoluble compound. Glycogen phosphorylase B is going to be the enzyme that catalyzes the reaction we see in figure C, which is the breakdown of glycogen to produce individual residues of glucose 6 phosphate. Ultimately, we study the effects of biotin on glycogen phosphorylase B by doing two main things: enzyme kinematics and absorbance spectroscopy. As we can see in the graph A, the phosphate concentrations and absorbance values, we modeled the absorbance at 600 nanometers versus the concentration of inorganic phosphate. Inorganic phosphate is going to be a crucial substrate used by glycogen phosphorylase B. With this link established, we can calculate the reaction velocities, concentrations of phosphate, etc., based off the A600 readings we acquire. So the main aim of this study was to establish the link between biotin and glycogen phosphorylase B activity. But before we actually added biotin to our assays, we needed a point of reference. So first we did calibration assays with varying concentrations of AMP or adenosine monophosphate as an effector of the enzyme. Adenosine monophosphates can be indicative of low energy levels and low glucose levels within the cell. So typically that molecule is going to act as an activator of glycogen phosphorylase B, as the molecule will then force the enzyme to increase blood sugar levels and allow glucose to travel throughout the body. It mobilizes the liver to break down glycogen and increase serum glucose levels. And we noticed that AMP acted as a activator of the enzyme. So with an unknown effect of biotin added to our enzyme, we carried out four assays. Two assays with high concentrations of biotin and varying levels of AMP, and two assays with low levels of biotin and varying levels of AMP, which, as we summarize in the table right here, the two concentrations of AMP we used were 4 millimolar and zero point 0.5 millimolar, and we used 0.0133 milligrams per milliliters of biotin for the high concentration of biotin and 0.0033 milligrams per milliliter of biotin for the low concentration of biotin. In looking at our graphs in our summary table in figure C and the biotin assays table respectively, we saw that ultimately biotin was an inhibitor of glycogen phosphorylase B at high concentrations of AMP, and actually almost counterintuitively led to increased levels of glycogen phosphorylase B activity when there were low levels of AMP present. However, due to kind of like the small margins between the Vmax calculated from our graphs, with all the Vmaxes reaching around 9 or 10 millimolars per minute, it's very difficult to differentiate and find a tangible effect without statistical and error analysis of biotin's activity and effects on glycogen phosphorylase B. So my initial conclusion is going to lead me to conclude that biotin served as an inhibitor of the enzyme at high AMP levels, but served as a actual activator of glycogen phosphorylase B at low AMP levels. Now, ultimately, we have to understand that this reaction in our study is going to be limited due to the fact that biotin has a crucial role in hexokinase activity as well. And hexokinase activity is going to increase the actual levels of glycogen synthesis, which would counteract glycogen phosphorylase B. So ultimately, there's still further study needed to be done to link the causality and the effects of biotin on glycogen phosphorylase B. then it's likely that this cofactor serves as an inhibitor of the enzyme.